Acquired immunity is pathogen-specific. It depends on the body's memory. For example, when a body encounters a bacterium for the first time, it produces a low-intensity response called a primary immune response and produces memory cells. However, when the same bacterium attacks again, the memory cells help to elicit a secondary immune response of high intensity. This response is also known as an anamnestic response. Both primary and secondary immune responses are made possible by two special lymphocytes in our blood, B lymphocytes and T lymphocytes respectively. B lymphocytes produce proteins to fight pathogens, whereas T lymphocytes help B cells to perform this task. The protective proteins produced by B cells are called antibodies. Each antibody molecule has 4 peptide chains, 2 small and 2 longer ones. The 2 small chains are called light chains, while the 2 longer ones are called heavy chains. Therefore, an antibody is represented by H2L2. IgA, IgM, IgE and IgG are some examples of antibodies in our body. Acquired immunity, or the acquired immune response, is primarily of 2 types: a humoral immune response and a cell-mediated immunity, or CMI. It is the humoral immune response that is carried out by antibodies. Since antibodies are found in blood, it is called the humoral immune response. The word humoral refers to blood or any other body fluid. The cell-mediated immune response can be best understood with the example of an organ transplant. When organs like the eye, heart, kidney or liver fail, organ transplant is the only remedy. Thus begins the search for a suitable, healthy donor for transplantation. This is important because grafts or transplants cannot be made from just about any source, as the body will reject those grafts sooner or later. To find a perfect match, tissue matching and blood group matching are conducted before a transplant. This ability of the body to differentiate between self and non-self while rejecting grafts is the cell-mediated immune response. It is an immune response that doesn't involve antibodies, but it involves the activation of macrophages, natural killer cells or NK, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines that fight antigens. Patients who have had organ transplants have to take immune suppressants all their lives to inhibit their body's immune response against the transplant. Then again, based on whether the antibodies are produced inside or outside the body, immunity can be distinguished as active immunity or passive immunity. When the body is exposed to antigens such as living or dead microbes or other proteins, it produces antibodies in response. This type of immunity is called active immunity, which is slow and takes time to reach its full effective response. When microbes gain access to the body during a natural infection, active immunity is induced. Consequently, the underlying principle of any immunization plan is active immunity. where it is induced by deliberately injecting microbes into the body. On the other hand, passive immunity comes into play when ready-made antibodies are delivered directly into the body from external sources to protect it against foreign agents. The antibody that a fetus receives from its mother through the placenta during pregnancy is an example of passive immunity. Likewise, during the initial days of lactation, mother's milk contains a yellowish fluid called colostrum that has copious amounts of the antibody IgA. Therefore, mother's milk also provides passive immunity to babies. Did you know that even though passive immunity provides immediate protection to the body against pathogens, the body doesn't develop any memory of the pathogen? This means that the body is at risk of being attacked by the same pathogen when infected by it later. Therefore, immunity can be innate or acquired. It can also be either active if induced by an infection or a microbial attack, or passive if provided with ready-made antibodies. The immunity of the human body is based on memory. When the body encounters a pathogen for the first time, it reacts with a primary immune response and produces memory cells. When the body is attacked by the same pathogen twice, these memory cells offer an intensified secondary immune response. This memory of the immune system is also used as a principle in the immunization program. A preparation of antigenic proteins of pathogens or an inactive or weakened pathogen is introduced into the body as a vaccine. The antibodies produced in the body against these pathogens are then able to neutralize the pathogens during an actual infection. Vaccines generate memory, which enables B and T cells to recognize the pathogen quickly and produce an army of antibodies in response. Sometimes, when a quick immune response is required, for instance during a tetanus infection where the body is infected with deadly microbes, preformed antibodies are injected into the body. It's the same with snake bites, where patients are injected with preformed antibodies or an antitoxin, which is a preparation of antibodies against the snake venom. This type of immunization, involving preformed antibodies, is called passive immunization. New technologies like recombinant DNA technology have made the large-scale production of antigenic polypeptides of pathogens from yeast or bacteria possible. The hepatitis B vaccine is produced in a similar manner from yeast. In higher vertebrates, like human beings, memory-based acquired immunity is so evolved that they can differentiate foreign organisms such as pathogens from self cells. Most experimental immunology is based on this aspect. In addition, sometimes due to genetic or other unknown reasons, the body attacks itself or self cells. This attack damages the body and is called an autoimmune response. An example of an autoimmune disease is rheumatoid arthritis that affects the joints. Similarly, apart from an autoimmune response, the immune response also comes into play during an allergy. It is interesting to note that some particles in the environment, such as pollen, dust, and mites, cause us to sneeze or wheeze for no apparent reason. This usually happens because people are allergic or sensitive to these particles. This exaggerated immune response to certain antigens in the environment is called an allergy, whereas the substances to which such an immune response is produced are called allergens. Dust, pollen, animal dander, and mites are some common allergens. The antibodies produced against these allergens are usually the IgE type. A runny nose, sneezing, watery eyes, and difficulty breathing are common symptoms of allergic reactions. An allergic reaction is caused by the release of chemicals like histamine and serotonin from mast cells. Therefore, the use of drugs like antihistamine, adrenaline, and steroids quickly reduces the symptoms of an allergy. To determine the exact cause of an allergy, the patient has to either be exposed to or injected with small doses of different allergens. The patient's reaction to each allergen is studied to arrive at a conclusion. Somehow, modern-day lifestyles that border on unhealthy have lowered natural immunity and heightened our sensitivity to various allergens. Moreover, a protected early childhood doesn't prepare the human body for the vagaries of the environment in a metro city. Therefore, even though our environment is continually getting polluted and we are exposed to thousands of antigens every day, it is a combination of the body's immunity and a vaccination program based on immunization principles that help us combat infections and diseases. The human immune system works as an armor against infections and diseases. It helps the body distinguish between non-self cells like bacterial, fungal, and viral pathogens from self cells. The immune system responds to non-self agents and remembers them. It also plays an important role in reacting to allergens, organ transplantation, as well as autoimmune responses. The human immune system consists of lymphoid organs, lymph nodes, and lymph cells called lymphocytes. The lymphoid organs are where lymphocytes originate, mature, and proliferate. Lymphoid organs can be classified as primary lymphoid organs and secondary lymphoid organs. Bone marrow and the thymus are two primary lymphoid organs. Bone marrow is a flexible tissue found in the hollow interior of bones where all blood cells, including lymphocytes, are produced. The thymus, on the other hand, is a lobed organ which is located near the heart and beneath the breastbone. The thymus, which is quite large at birth, shrinks in size with an increase in age. By the time a person reaches puberty, the thymus shrinks considerably. Both bone marrow and the thymus provide the environment for the development and maturation of T lymphocytes. It is in these two organs that immature lymphocytes differentiate into antigen-sensitive lymphocytes. When lymphocytes mature, they move to the secondary lymphoid organs such as the spleen, lymph nodes, tonsils, Peyer's patches in the small intestine, and appendix. It is inside the secondary lymphoid organs that lymphocytes interact with antigens and proliferate to form effector cells. The spleen is a large bean-shaped organ located in the upper left part of the abdomen, protected by the rib cage. It chiefly contains lymphocytes, erythrocytes, and phagocytes. It acts as a large reservoir of blood to be supplied in times of emergencies like hemorrhagic shock or excess loss of blood due to cuts or injuries. It also plays the role of an RBC graveyard by removing old or damaged RBCs from the body. Apart from the lymphoid organs, the human body has clusters of lymph nodes in places such as the neck, underneath the arms, and in the groin. Lymph nodes trap microbes and antigens that enter lymph and tissue fluid. It is these antigens trapped inside the lymph nodes that activate the lymphocytes to produce antibodies, resulting in an immune response. Another lymphoid tissue called mucosal-associated lymphoid tissue, or MALT, is located inside the epithelial lining of major tracts like the respiratory, digestive, and urogenital tracts. The T cells, B cells, and macrophages present in MALT help regulate mucosal immunity. MALT constitutes about 50% of lymphoid tissue inside the body. Therefore, the immune system, which collectively comprises the lymphoid organs, lymph nodes, and tissues, protects the human body from invasion by pathogens and